If you haven't signed in at some point, make sure you do that. Is my braces? Is that, okay. We're going to go ahead and start with a prayer. This is a prayer for our seniors. It's a little corny, but you guys are corny. In the name of the Father, the Son, the Holy Spirit. I have knowledge, so you will show me now how to use it wisely and find a way somehow to make the world I live in a little better place and make the life with its problems a little bit easier to face. Grant me faith and courage and put purpose in my days and show me how to serve in the most effective ways. So my education, my knowledge, and my skill may find their true fulfillment as I learn to do thy will. And may I ever be aware in everything I do that knowledge comes from learning and wisdom comes from you, Lord. We ask this in your name, O Lord. Amen. Father, Son, Holy Spirit, amen. If you noticed the program, we have quite a few presentations, a little more than we've had in the past. This is a really tight-knit group, but when it came to the presentations and doing their projects, they were really independent. And they kind of scattered. And so we got them back together for 2 nights. And so I think, and quite a variety of projects as well. So we're going to go ahead and get started. Before that happens, though, I also wanted to recognize the mentors that are on the back. We have quite a few. And same thing for next week. These kids went out and found some people to help them out as best they possibly could. And so we definitely couldn't do that without their assistance. Some of the folks we've had before, and kind of regulars, Dr. Marianne Elliott from WSU, and she's always there to help us out. And we have a few new folks that maybe we can keep their relationship too. So, in any case, we're going to get started. The kids we're going to kind of roll them out since we have so many. We'll do 3 and then take a brief break. Give you a chance to kind of stretch, do 3 more, take a brief break, and then finish up. So Vasanth is going to be our first presenter. Come on up. Oh, you don't like when I pronounce your name. That's right. Sorry.
All right. Hello, everyone. My name is Vasanth Ramachandran, and my project is on examining the trainability of VO2 max between men and women ages 45 to 60. So just a brief intro of VO2 max. It's a chief measurement of an individual's maximum rate of oxygen consumption during exercise, and it's often used as a primary indicator of cardiorespiratory fitness and overall heart health. A higher VO2 max is associated with a reduced risk of diabetes, cardiovascular disease, and hypertension. So on the chart here, you can see that the highest 20% of VO2 maxes for men and women are at 37% and 39% less risk for cardiovascular disease, respectively. So moving on, much of the prior research regarding VO2 max has been done in younger populations, usually between the ages of 18 and 35. Adding on, so the chart below here shows the VO2 max norms for men and women, indicating that men generally have higher VO2 maxes than women. However, there is a lack of research regarding the ability to train VO2 max, and also among persons that are like middle-aged or usually between the ages of 45 to 60. There's also a lack of research regarding the trainability of VO2 max between men and women, and basically whether or not one gender can improve their VO2 max greater than others. So I decided to examine the trainability of VO2 max between men and women in the ages of 45 to 60 when prescribed the same dosage of aerobic exercise training. To examine the trainability of VO2 max, I chose a total of 8 participants with 4 men and 4 women. So if you look at the chart, I kept age, training, and altitude the same, but then I changed up the gender. My hypothesis was that men would see greater improvement in VO2 max over a 12-week regulated training program compared to women, because men usually have lower body fat percentages, higher muscle mass, and a higher hemoglobin concentration, which aids with blood flow to the muscles. This would thus lead to greater VO2 max. So for the methods, 4 men and 4 women between the ages of 45 to 60 were recruited. They were selected based on multiple factors such as age, gender, based on aerobic fitness levels, and previous exercise habits that were assessed through the International Physical Activity Questionnaire, or IPAC for short. For reference, the participants chosen considered themselves to be quote-unquote fit and had habits of exercising 4 to 5 times per week. Apart from these requirements, participants were also required to be free of use from heart rate lowering medications, because being on these would result in inaccurate measurements. So during the intervention period, I used 2 point systems to maintain a consistent training volume. The Personal Activity Intelligence, or PI point system, was employed in the study, and basically what it does is it gives a computerized and personalized score based on the subject's height, weight, heart rate, and duration of exercise. So essentially, the higher the heart rate, the more PI points one will earn. It offers a weekly summary score every Sunday, which is the number that you see in green there. Besides that, I also used the Cooper aerobic point system. The Cooper point system works more on the type and the duration of exercise, with more points for a longer workout or a shorter but more intense workout. So it's a little tough to see, but here you can see you get 5 points for running 1.5 miles in 15 minutes, but you also get 5 points for running 1.2 miles in 12 minutes. So as for the study itself, it would be a 12-week study with the first 4 weeks on the Cooper point system and the last 8 weeks on the PI point system. This was due to the fact that the PI app was actually discontinued after the first 4 weeks of the study, so we had to pivot for the final 8 weeks. When using the PI point system, participants were instructed to gain a minimum of 100 points per week. This was based on research from the Hunt study, which found that 100 PI points per week leads to a 30% decrease in mortality due to cardiovascular disease. In the Cooper points portion of the study, participants were instructed to get 30 Cooper points per week, based on research from Dr. Cooper, which found that 30 points per week led to increases in physical fitness and cardiovascular health. So moving on to the pre- and post-study procedure. Both before and after the 12-week study period, participants performed the Kenneth Cooper 12-minute walk-run test. This test essentially determines how far a participant can travel in 12 minutes and is usually performed on a standard quarter-mile running track. The test also offers for an accurate estimate of VO2 max using the equation 35.97 times the miles covered in the 12 minutes minus 11.291. So as a result, this test was the main way to obtain VO2 max measurements throughout the study. The participants were also given a post-study survey that I adapted from one originally made by UC Berkeley, and it was gained to get a handle on their physical, emotional, and mental well-being after a rigorous 12-week exercise schedule. Adding on, the participants tended to favor exercises like running, walking, and biking, more aerobic exercises, compared to more anaerobic exercises like weightlifting. Also, as you'll see in the results, one of the participants did have to drop out, so we were left with three women and 4 men. So moving on to the results, here's a graph showing the weekly PI points that each person got. F1 is female number 1, male 1 is male number 1, and so on and so forth. You can also see their weekly, or their 4-week average, which is the orange number right here. And as you can see, everyone did clear the 100 PI point minimum, albeit some cleared it more than others, as you can see there. This one depicts the average, or the weekly data for each person's performance during the Cooper points system. And it also offers their 8-week average, as you can see here in yellow. Again, each participant did clear the 30-point minimum threshold, albeit some did clear it more than others. Another thing to note is that some participants, they might not have cleared it every week, but then they would bounce back with a stronger week the next week to kind of even out their performance. So these 2 graphs represent the average weekly training volume comparing men and women. So the graph on the left displays the average weekly Cooper points for males and for females, and then this one, the average weekly PI points for males and for females. So you can see men on average did receive greater point totals during both the Cooper points and the PI points phases. And then I statistically analyzed the differences in both scenarios using a t-test. So for reference, a t-test, if you get a p-value of 0.05 or less, then it's considered to be statistically significant. So for Cooper points, the p-value return was 0.78, and then for pi points, it was 0.47. So neither was considered to be a statistically significant difference, although there was a little bit greater difference with the pi points. So here's a key graph here. This graph represents the average distance or difference in VO2 max between genders, which is the focal point of the study. And according to the graph, both groups saw nearly identical changes to their VO2 max, with men seeing a 5.32 milliliters per kilograms per minute increase, and women at a marginally higher 5.53 milliliters per kilograms per minute increase. So according to the graph, there's really no difference in the difference. There's no real difference in the change of VO2 max between genders. This is also backed up by the t-test that I did, which returned a p-value of 0.915, so it's almost like identical. So moving on to the discussion, the main takeaway is that in my sample of participants, there was no significant differences in the trainability of VO2 max, so this rejects my hypothesis that men would see a greater increase, but it does support the null hypothesis that there would be little to no difference in VO2 max change. The differences in training volume as assessed by the Cooper and pi point systems proved to be basically irrelevant, and it wasn't enough to cause any market changes. My data, however, was kind of consistent with previous research that found that men usually have a 15 to 25% higher VO2 max than women. In my case, they were right around 15% higher than women's. This is again due in large part to the fact that men have higher muscle mass, lower body fat, and a higher hemoglobin concentration. I couldn't measure hemoglobin concentration, but the men in my study did have lower body fat and higher muscle mass percentages. So moving on to some of the anecdotal responses that I got from the survey. The hardest part for the participants was to stick to a regimented workout routine and maintaining that discipline. They did report, however, that performing a smaller set of exercises done with greater frequency made it a lot easier to kind of stay consistent. They also reported that maintaining a strict diet was made easier when exercising because they didn't want to, kill their gains that they made by exercising by eating unhealthily. They also reported that they felt less mental fatigue and greater positive thoughts through exercising, which is to be expected, obviously, because exercising releases endorphins. So basically, what we learned here is that if you're really struggling to maintain an exercise schedule, it's better to perform a smaller set of exercises that you can do really frequently so that you can kind of harness the benefits of your exercise. So obviously, there were some major limitations with this study. The most prominent one being the fact that it was a pretty small sample size with only 7 participants. As a result, it is kind of difficult to gain a concrete understanding of the magnitude of trainability of VO2 max, and it's kind of unclear if the insignificant differences are indicative of a trend of the wider population or if it's just kind of localized to mine. Another significant limitation was the fact that I didn't have access to VO2 max, like official VO2 max testing equipment, because this is only available to medical professionals in hospitals, and I'm not a medical professional, so yeah. The tests that we did use, they're widely regarded as being highly accurate, especially the Cooper test, but at the end of the day, they still are estimates, and it's not going to be as accurate. However, there are definitely avenues for further research, the main avenue being to repeat the study with a greater number of participants. You can either keep the same demographic or you can change up the demographic based on age or starting fitness level. Another avenue is obviously to repeat the study with access to VO2 max testing equipment, so kind of in a more clinical setting, especially for the pre and post tests where you can put the participants on the treadmill and measure their VO2 max. So yeah, I just have a few acknowledgments to make. Firstly, I'd like to thank my mentor, Mr. Glenn Bean. I know he's not here today, he's kind of sick, but he's been a tremendous resource for me and friend throughout this whole process. So thank you, Glenn. I'd also like to thank my parents for keeping me on track and being supportive throughout the whole study, always nudging me when they thought I was being too lazy. The participants of the study also deserve a huge thanks because they maintained the discipline to complete the whole 12-week study, and it wouldn't have been possible without them. Also like to thank Mr. DeGroat and Mr. and Mrs. Nielsen for the opportunity to be in this wonderful program. And last but not least, to this year's Marine Chem class, it's been great to get to know you guys. We're excited. Are there any questions? Yes, Mr. Nielsen. Which one? Okay. This one? This one? Yeah. Yeah, so male 4 was actually in the Army for like a long time. So he was used to running like 7 miles a day every morning. So he would just send me his 7 miles at like 6 a.m. and be like, you know, I already did my running for the day. I wouldn't have even been up. So yeah. So his improvement though, because he was he was already kind of used to running that 7 miles, he did see improvement, although it was on par with basically everyone else, as it wasn't like an outlier or anything like that. Any other questions? No? Okay. All right. Thank you.
Hey, I don't know if people knew this or not, but we are streaming this. And so you can get on your phones right now and tell all your people. I'll give you the link to that. I think we're already up well over 10, 12,000 people probably watching this right now. I anticipate over 100,000 by the evening. So, all right. Sophia, you're up next.
Preview. Okay, I'm Sophie Nicholson, and my project was testing the effects of blue light and stimulus on sleep quality and quantity in adolescents. So just let's start with a raise of hands. How many people think they don't get enough sleep? Yeah, that's kind of what I thought. According to the American Academy of Sleep Medicine, adolescents 13 to 18 should sleep 8 to 10 hours a night, and adults should get 7 to 9 hours. And some people may doubt this, but there are a lot of studies that back this up. So sleep has many different purposes for the body, from modulating hormone levels, metabolic status, and the immune system, to strengthening memories during REM sleep and eliminating unnecessary brain connections. Creative thinking also seems to be affected by sleep loss, and multiple studies have found a positive correlation between less sleep and poor academic performance. So generally they found that students with less sleep had lower GPAs. So in the short term, lack of sleep can lessen a student's learning capabilities and general academic performance. So blue light is generally defined as light within about 415 to 530 nanometers, so short, high-energy wavelengths. It's found in high quantities in daylight as well as in light from devices. Melanopsin, a photopigment found in our eyes, is a receptor that signals for the creation or suppression of melatonin, which is a hormone produced by our bodies in the pineal gland. Melanopsin is triggered by specific wavelengths of light and is most sensitive around 480 nanometers, which is also in the range of blue light. When melanopsin receptors absorb blue light, they suppress melatonin production. So melatonin release levels change throughout the day, less when you're exposed to light in the morning to help with alertness, and more when it gets dark to help with sleepiness. Because of this, blue light can play a big role in affecting sleep. But how big is this effect? Studies have shown that even a small amount of blue light can suppress melatonin. Blue light helps to synchronize the circadian rhythm throughout the day, but it causes large problems when you're exposed to large amounts late at night. And the CDC has found that adolescents 15 to 18 generally spend about 7.5 hours in front of screens every day. Less is known about the effects of visual stimuli, though one study showed that there may be a correlation between visual stimuli and the disruption of a person's biological clock. A plethora of studies have been focused on the importance of sleep and some on the effects of light or blue light. Few have looked deeper into what the biggest effect on sleep is, though. Kane et al. Discovered a negative correlation between media usage and delayed bedtime or shorter sleep time. Lamola et al. Discovered electronic media usage was negatively correlated with sleep length and increased sleep difficulty. So I was curious, is it the blue light, the stimulus from the screens, or both? And as I researched further, I found the study D'Orazio et al., which I drew some inspiration from. In their results, they found no meaningful difference for the sleep data between groups that used their phones like normal and groups that had a blue light filter on their phones, but they did find that a group that didn't use their phones for an hour before bedtime had increased sleep. So to get some background information, I sent out an initial survey just to get an idea of who would be willing to participate in the survey, as well as to get an idea of the typical Bellarmine high schooler's sleep profile. So 91 answered at least one question, 32 enrolled in the Shobi class at the beginning of the study, and 16 participated consistently, with 14 participating in all stages. Around 54% of the students who answered the first question about how much sleep they got reported getting less than 8 hours of sleep as a general rule, and 76% of those who answered the question about when they shut their devices off before bed reported shutting their devices off less than half an hour before bed, which logically should have an effect on melatonin levels. So my question was, what are the effects of blue light and visual stimulus on sleep, and which is more disruptive? While my study had a similar base to the D'Orazio study and both had similar aims, I changed a few key pieces. First, I studied high school students ages 14 to 18 instead of adults 18 to 24. In addition, participants took part in each stage instead of dividing them up into 3 groups and assigning them a specific stage. Also, for feasibility purposes, the students tracked their sleep with an app instead of a wrist-worn tracker, both to increase the populations of students I could pull from and for cost purposes. I also added an initial survey, like I mentioned, and a conclusion survey. So I decided to make each person their own control instead of just dividing participants into groups, because each person has a very different sleep schedule. This resulted in a 9-week-long 3-stage study, and the free app Sleep Time Free was used to track sleep every weekday night, turning in screenshots every weekend into Shobi with periodic reminders throughout the week. The first stage was the control, where each participant tracked their sleep with the earlier parameters with no change to their normal sleep routine. Then in the second stage, participants were asked to turn on Apple Night Shift to the warmest setting for the entirety of the 3 weeks. So Apple Night Shift shifts the light emitted from devices to the warmer end of the spectrum, negating the effects of blue light while leaving the visual stimulus, and tracking continued as normal. In the last stage, participants were asked to refrain from using their personal blue light devices for 30 minutes before bedtime. This was a shift from the D'Orazio study's hour, both out of curiosity over whether it would make a difference and because of necessity, because high school students voluntarily participating were highly unlikely to stay off their phones for more than half an hour before bed. In addition, there was a short survey at the end of the study to assess qualitative differences as well as self-reported accuracy. The pictures here are an example of a relatively normal or good night's sleep. The orange is awake time, the blue is deep sleep, and the green is light sleep. So as the study concluded, I entered each person's data into a Google spreadsheet, converted the data into hours, and then used Data Classroom to analyze it. So total time is the time that each participant spent in bed, so from when they started the sleep tracker to when they stopped it. And you can see that on the right. And then the actual sleep time is the time that the app recorded the participants as asleep, which is on the left. So originally I organized data for each week, like shown here, but after working with it, I decided it made a lot more sense to organize it by stages and groups of week, especially with so many individual data points. So in the end, like I referenced earlier, 16 participants consistently tracked and turned in data, with 14 participating in all stages. When the data was analyzed with an ANOVA test, no statistically significant difference was found throughout the stages. However, there was an increase in actual sleep time from the first stage to both the second and the third, increasing 0.297 hours from stage 1 to stage 2 and 0.29 from stage 1 to stage 3. There was an increase in total sleep time as well from the first stage to the other two, increasing 0.288 hours from stage 1 to stage 2 and 0.337 hours from stage 1 to stage 3. There was a very slight difference between both the total and actual sleep data for stages 2 and 3, but there was no discernible pattern. I also compared data by pulling out different weeks to see if there was a statistically significant difference between the beginning and end of each stage or between any groups of weeks. And interestingly enough, when comparing weeks 3, 6, and 9, so the last weeks for each stage, I did find that there was a statistically significant difference between weeks 3 and then 6 and 9 for total time, and between weeks 3 and 6 for actual time. The actual time difference increased about an hour from weeks 3 to 6 and about half an hour from weeks 3 to 9. The total time difference increased about 2 hours between weeks 3 and 6 and about an hour 0.73 between weeks 3 and 9. However, this statistically significant increase was not reflected in any other week groupings. So in the end, there was a slight increase in the time in bed and the time asleep from the control stage with normal habits to both the blue light filter and the no device stage. However, similarly to the results from the Durascio study, the difference was not statistically significant, likely partially due to confounding factors outside the study's limitation, as well as the relatively small sample size. There was no pattern at all in the difference between the data from stage 2 and 3, though, which was different from the Durascio study, which found that the no device stage had a bigger impact than the blue light filter stage. However, together with that study, there is some evidence that not using phones right before bed can make a positive impact on sleep. Another interesting discovery was that 9 out of 12 participants who responded to the final survey indicated that they believed one or more of the stages helped with their level of alertness or time it took them to fall asleep, even though the data didn't necessarily support this, leading to the conclusion that simply paying more attention to their sleep habits or changing them made them believe they were better rested. The statistical significance between weeks 3, 6, and 9, while interesting, was not reflected in any of the other week groupings and therefore likely by chance. So there are some limitations with this study. First, possibly in the app that tracks sleep. As you can see here, these pictures look a bit different from the first sleep pattern picture, much more time spent awake or in light sleep. The app uses different algorithms to differentiate between cycles of sleep, so light sleep and deep sleep, based on movement levels. However, it's possible that this data was skewed depending on where the participant placed their phone in relation to them and whether they were moving it. However, as unusual as this data looked, for some people it was interesting to discover that the patterns for the actual and total sleep time were the same. It also would have been interesting to expand on the surveys to look further into whether participants thought that they were waking up a lot and to have an identical study to compare from the beginning to the end. The main source of error, though, likely came from the fact that as a voluntary study, I depended on participants turning in their data consistently in order to use it, which limited my ability to use much of the data as participants did not track consistently. In addition, 4 out of 12 participants who responded to the final survey indicated that they did not follow the no devices within 30 minutes before bedtime guideline. In addition, without having students record what they ate and when and what they did all day, it was impossible to control for all possible confounding variables, such as stress levels, total caffeine intake, and late exercise that could affect sleep. So going off this, there are many possibilities for future research, especially with all the confounding variables. First, all the participants who answered the concluding survey indicated that they did not watch TV in the interval that they were asked to be off their devices. It would be an interesting follow-up to examine whether there were sleep differences, whether the student watched TV or used their devices before bed. It would also be a possible study to do this similar study, but also using wristward trackers and an app to see if the data was similar. Lastly, it would be interesting to study whether other variables such as caffeine, stress levels, and late exercise compared to these devices in terms of sleep impact. And we have another science fair coming up, so I'll be continuing to analyze this data, specifically looking for trends in whether there were instances of waking in the night in different stages more than others. And lastly, just acknowledgements. I wanted to thank my family for always supporting me, even when I refused to give them specific details on what I was doing, and to Mr. DeGroot for helping me in Hawaii with diving, as well as with all my statistics and trying to figure out my project. And of course, real Neilmobile, obviously the best van. And the Marine Chem class of 2023, and of course to my participants, especially those final consistent 16. So, thank you. Okay, any questions? Okay. Hayden, Curran. Yes? No, so this was going from around October to December before Christmas break. So I didn't do it over the break just because I knew that sleep time obviously increases over breaks, so I made sure to keep it within that time frame. Did you have a question? Yeah. I don't, sorry. Since it was before Christmas break, do I think midterms made a significant difference in the data? I'm guessing not, because there wasn't, if anything, it'd be likely because midterms were right before break that the data should go down, the sleep amount should go down. And while, like I said, there wasn't a statistically significant increase, there was an increase. So I'm guessing that did not affect that like that. Yeah. Okay. Yeah, Mr. Nielsen. Okay. Which one? This one or the other one? Do you mean like, can I see each individual participant, or? Yeah. Sorry, what's your question exactly? Okay. Yeah, that is, so they did record there were a couple of people that consistently got a very low amount of sleep. So that's, yeah, that is what the data was. Pretty much. It was at least in that lower range generally, yeah.
This is based on what they received from the app.
Yep. Okay.
Hi everyone, I'm Madeline Cragen, and this is my Marine Chem project, genotyping of novel phytochrome mutants in Solanum lycopersicum, or tomato plants. So we're going to start by talking about photosynthesis. So as we all know, photosynthesis combines carbon dioxide and water to make energy for the plant and oxygen. But another key factor in this is sunlight. And so that's really what my project deals with. And so phytochromes are the plant's light receptors, and they function like the eyes of the plant, so that they're similar to human eyes and that they take in certain wavelengths and process them. And so the phytochrome is made up of a chromophore, which is this little blue square, which is what's specifically responsible for taking in the light, and an apoprotein, which is this protein chain that's chemically bound to the chromophore. And so that molecule called a phytochrome is what takes in light for the plant. And like human eyes, phytochromes only can process certain types of light. And so while the human eye only processes light from the visible spectrum, phytochromes process red and far red light. And they're considered open for far red light, which means they're active and able to grow, and they close for far red light. And at different times of the day with the changing levels of red and far red light, they either open or close to absorb sunlight and grow. And so specifically in tomato plants, there are 5 types of phytochromes: Phi A, Phi B1, Phi B2, Phi E, and Phi F. And so far, most of the types of phytochromes have been pretty widely studied, but Phi F was actually discovered in the Mad Lung lab where I did my project, so there isn't as much research about it. And so that's part of what I was looking into, is the effects of Phi F. And so the way that we study phytochromes is that we look at mutations, because they're what's known as a knockout mutant. And so the functions of the phytochrome are rendered just completely functionless when there's a mutation. And so when we look at mutations, we're able to compare a plant with some sort of phytochrome mutation with a regular plant. And we can breed specific combinations of mutations to understand more about the interactions of these mutations. And so here we are looking at a plant that has some sort of mutation here and a regular plant. And so even though we don't necessarily know the function of the phytochrome that's missing in this plant here, we can see that it's some sort of function that has to do with plant growth. And so even if we can't necessarily determine the exact function, we can see some of the functions when we know what phytochromes are missing. And so the way that we kind of look at this is we use gene segregation. So this is just a basic Punnett square for 5B1. And so here we know that the parent plant up here is carrier of a mutant. The parent plant on this side is also a carrier of the mutant. And because they're both heterozygous, there's a 1 in 4 chance of getting this B1 mutation. But in nature, the chances of this happening are a lot lower because we don't know the genotypes of the parents, and they're not usually a mutant, which is why we study them in the lab to try to understand what these mutations mean and the functions of these phytochromes. So I looked at all 5 phytochromes. So this is a little bit more complicated, but it's functionally the same thing. So my plant was homozygous dominant, meaning it just had the regular gene for Phi A and Phi E. It was heterozygous, meaning it had the wild type and also carried the mutant for Phi B1 and Phi B2. And it was homozygous recessive for Phi F, meaning it was already a mutant. So this was the overall genotype. And so I cross-pollinated these plants, which means that we took pollen from the plant and put it in other flowers to create seeds. So the parent was the same for both the top and the side. And so with this genotype of the parent plant, we had a 1 in 16 chance of finding this triple mutant right here that you see with a Phi B1, Phi B2, and Phi F mutation. So that leads into my research question. And so my goal was to isolate this triple mutation for Phi B1, Phi B2, and Phi F so it can be studied for future research about what this mutation can mean. And so using PCR and gene sequencing, I believe that I could find this mutation. So starting off by talking about just what a polymerase chain reaction, or PCR, is so it's a combination of combining primers that bind to the DNA to isolate a certain section, and the DNA goes through a process of heating and cooling in this PCR machine, which replicates the DNA strand. So we start with 1, then to 2, to 4, to 8, and keep going until we make millions of copies. So we have a really small section of this DNA that we can look at really closely to understand what the genotype is and whether or not it's a mutant. So starting off with B2, for this procedure, I used a gel. And so that was where I put my PCR product into these wells up here. And so the non-specific is just telling us that there is DNA present for that plant. And then we use the wild type and mutant bands to determine the genotype of the plant. And so if we've had a non-specific band and a wild type band, that tells us that we have a wild type, or it's homozygous dominant, meaning it just carries the regular gene. If we had the non-specific band and the mutant band, that tells us that it's a mutation, or it's homozygous recessive, which is what I was looking for. And then if all 3 bands were present, it's heterozygous, which was the original genotype for B2. And that means that it carries the mutation, but it also carries the wild type. So that wasn't what we were looking for. So this was what some of my actual results looked like. It's not as pretty as the drawing, but it tells the same story. So plant number 7 was heterozygous. You can see the nonspecific, wild type, and mutant band. Plant number 8 was wild type. We have just the nonspecific and wild type. And then here you see plant 10 has the nonspecific and the mutant band, meaning this was a mutant. And so I ended up finding 2 wild types, 10 heterozygous, and 3 mutant plants. And so plants 2, 6, and 10 were mutants. And so once I had determined this, I needed to study those for B1 because since I knew they were already double mutants, I just wanted to see if they were triple mutants. So I only studied B1 for those 3 plants. So for B1, the procedure is a little bit different because B1 is only a single base pair mutation, which we'll talk about later. But B2, there's a whole chunk missing, so it's easy to see on a gel. But we can't isolate that small section with primers for B1, so we have to genotype the whole thing. So here I just ran a PCR that's a similar function to a non-specific band. It just tells us that DNA is present. So here are these were the 3 wells up here. Let's see, this one, and this one. Plant 2, 6, and 10. So as you can see, no DNA was present for B1 for plant 2. So I didn't end up sequencing that one, but I did sequence plants 6 and 10. And these were my results for the sequencing. And it looks complicated, but I promise you that it's not. This top strand is just regular DNA that we have on file in the lab that tells us what a regular B1 sequence looks like. And then below here is what my B1 plants look like for both plant 6 and 10. And as you can see, there's a single base pair mutation right here from a C to a T, which tells us that the B1 mutation is present, but that doesn't tell us necessarily if it's heterozygous or fully a mutant. And so to understand this, we have to look at the trace file, which is this funky looking graph down here. And so if you see this black line, that's just to grab your attention to see that this spot in the graph. And so this is where that mutation would be. And so you see there's a single peak right here. And if it were to be heterozygous, there would be a double peak, meaning that not just the mutant, but also the wild type was present. But since only the mutant was present here, that confirms that this was also a B1 mutant. So I did find that both plants 6 and 10 were triple mutants for 5B1, 5B2, and 5F. So I was able to find what my research question set out to do. And so the only limitation I really had was the problem of DNA degradation. So as I told you earlier, plant 2's B1 PCR degraded, so I wasn't able to sequence it, but that didn't end up being a problem. But also earlier in my research, the B2 DNA for plants 1 through 5 degraded because I did those first, and then when I put them in the gel mold, nothing showed up, so I had to re-extract DNA from those plants and rerun the PCR in the gel, but I did end up getting regular results for those. So it wasn't too much of a problem. And so now, these are pictures of my plants. I took them about a week ago growing in the greenhouse at UPS. And so right now, these are offspring of plant number 6. And so they're undergoing light treatment now. So some are undergoing constant red light, some are undergoing constant far red, and some are undergoing light pulses, just to do further research to understand more about what these mutations mean. And so even though from this study I couldn't determine exactly what this mutation tells us, I can speculate about what it could potentially be for past knowledge about a B1, B2 double mutation, because that has been further studied. So if the triple mutant had similar problems and functions to a double mutant, that would tell us that 5F has similar functions to 5B1 and 5B2. But if this plant had some sort of different problem or different function, that could clue us into what the 5F function is, because if that's lacking in the triple mutant, then we know that that's part of the function of 5F. So while more research needs to be done to fully determine this, we can see some of the results now. And so not only can this phytochrome research be used just to understand more about how plants work, it can also be used if there was some sort of environmental problem with tomato plants in the future, because as we all know, tomato plants are super important for agriculture. And so if there were ever some problem with growth, we could potentially look to a phytochrome mutation to explain this problem. And with this research being done now, we could potentially fix it in the future if this were to come up. So even though I'm not fully able to say what this mutation does or what all the functions of the phytochromes are, it is important that this was found, because now we can do further research to understand what this mutation means. And so I'd just like to say thank you to Mr. DeGroat for being an awesome mentor in this program. I've had such a great time. Thank you for the Nielsen's for taking us on the Hawaii trip, and the Mrs. Neilmobile van is definitely the better van. And thank you to Dr. Andreas Madlung for letting me work in your lab at the University of Puget Sound. It was seriously such a great experience to get real lab work done, and I learned so much. And thank you to Christina Wynn, who was my undergraduate student mentor at UPS, for helping with my project. And to Sam Barnwell, our lab technician, who also helped me a lot in doing my research. And of course, thank you to the whole Marine Chem class of 23. I've had such a great time with you guys. And thank you to my family and friends for always supporting me and for supporting me through this project. Does anyone have any questions? Yeah? Oh, the question is, where did I extract the DNA from? So I took about 5 leaves from each plant, and then I froze them in liquid nitrogen, and then I crushed them up, and I added buffers and like primers to them to make the PCR product. Yes. So I knew that I had an initial idea about what I wanted to do some sort of plant biology project, and originally wanted to do something more climate change based. And so I was doing research about mentors in the area, and I found this lab, and it just seemed like the perfect opportunity. And so I reached out to the professor, and he responded, so it was pretty cool. Okay, thank you.
Hey, why don't we just take just a couple minutes if you need to stand up and stretch for a second, and then we'll get back in about 2 minutes or so. Okay, I hate to interrupt things, but we're going to go ahead and get back on the road again. And Mira's going to tell us a little bit about her research. Mira. Oh. Okay, then this is a good chance for me to embarrass her, maybe. Mira actually got started with her project the end of her sophomore year into the summer, so as a rising junior, and worked over the summer. It was right in the middle of COVID, and so she actually came to me after the summer and said, "Hey, I've got this project. I collected all this data, and what do you think?" And so she continued on with that, but I just needed to show you her publication that's in the International Journal of high school research, just came out in October. So this is based on her first study, and so what she's going to present is the follow-up to that. Does that mean another publication or something? Yeah, okay. Okay.
Okay, fair warning, I do have white slides, so this might hurt your eyes. Okay. Hi, my name is Mara Coons, and I'm doing my research and presentation on the impact of COVID-19 on adolescent anxiety and depression during the return to in-person learning. To give a little background knowledge into the impact of COVID-19, I'd like to start off by saying that the coronavirus has majorly impacted the world and caused a widespread concern that the mental health in children and adolescents has severely been negatively impacted due to the pandemic. In recent studies being conducted, they have also reported that COVID-19 has indeed affected adolescent mental health due to the lack of face-to-face connection and social isolation. Therefore, my research question is, how has teens' mental health changed since the return to in-person learning? Prior to conducting my survey for the project, which is what Mr. DeGroot was just talking about before I came, I conducted another survey back in the summer of 2021, which analyzed how COVID-19 has impacted adolescent mental health, specifically targeting social isolation and online learning. My results from that survey showed that there were significantly high levels of anxiety, depression, and stress throughout the majority of Bellarmine students during that time period. Some free responses even said that because of the little or no face-to-face interaction, they started to become more anxious and depressed. For this project, I wanted to compare my results from the summer of 2021 survey to results of the fall of 2022 survey to see if there are any changes in mental health within the same Bellarmine population. To target my research question, I made sure that the target audience were high school students in my region of Tacoma, Washington, ranging from 14 to 18 years old. Due to my limited abilities of traveling outside the region for more results, Bellarmine was the only high school I was able to survey. I used a validated survey called the DAS-21 survey to assess participants' mental health symptoms during the transition back to face-to-face education. This survey consisted of 21 questions and an optional short answer question, which allowed them to evaluate their mental health more deeply if they wanted to. Going deeper into what the DAS-21 survey is, it is a scale adapted from the DAS-42 scale, which is designed to assess the following subcategories: depression, anxiety, and stress. During the survey, the participants are asked to choose from 1 to 4 to help indicate how much the question relates to them. The higher the score in each subcategory, the more severe the symptoms are for that specific subcategory related to the respondent. We can see how severe each answer is based on whether the respondent subcategory is characterized as mild, moderate, severe, or extremely severe. After the respondents answered, I further analyzed my research on an Excel spreadsheet where the mean numbers were then calculated. I also gave a photo of what the DAS-21 survey looks like from a medical perspective, like if you're getting a checkup at the office. And it has similar questions, you probably can't see it from there, but of what my survey will look like, or did look like when I did send it out. I also wanted to show the differences and similarities between the two different surveys so you guys can see how even though the surveys may look a bit different based on the color or design, they are fairly similar. The main change between both of them is the way the participants thought about the survey, however. For example, for my first survey, I asked the participants to rate how much their mental health had changed due to social isolation and online learning. In my second survey, I asked the participants how the students' mental health has changed as they transitioned back to face-to-face education. In these tables, we see that each subcategory of stress, anxiety, and depression are broken up into, and which question is in each category. I also put the mean numbers calculated from each question in the table to see the differences between each question being asked and answered. Over here, you can see that this is the stress table, the anxiety table, and the depression table. In this table and figure, it shows the summative scores of each subcategory based on the results of my survey back in the fall of 2022. In the table, I showed the mean subcategory scores and which DAS-21 severity rating it got. In the figure, I showed the scores on a bar graph to contrast the differences between each subcategory. For the depression score, it got 34 for the mean subcategory score and extremely severe for the severity rating. For the anxiety score, it got 28.6, and it was also extremely severe for the severity rating. And for the stress score, it got 34.2, which is also extremely severe for the severity rating. After doing my research, I found out that the majority of respondents had stress, anxiety, and depression levels in the severe to extremely severe ranges. It is important to note, though, that the scores compared from this survey and the survey taken back in 2021 seem to differ. Going into the free response questions, I also found out that the majority of respondents felt alleviated when coming back to school and their moods elevated. I share some examples of the respondents' answers below. One example is, I felt around the second semester of 2021 and 2022 school year that I finally opened up and talked to people again. Soon after this, I was in a much better mood and saw that so many others had similar struggles as me. Another example was, a couple months after the return to in-person learning, I felt my mental health get better and my mind get more optimistic. I'm pretty sure it was from more social interactions in person. Based on other research being done by institutions such as UC Davis, my results seem to coincide with other research being done in the same field as mine. There is a decrease in anxiety and depression within students when returning back to in-person learning. As a conclusion from my original research question, though, it is supported, but there was an increase in stress compared to my survey back in the summer of 2021. But it also is important to note that my survey in 2021 did have higher levels of depression and anxiety scores compared to my data that I got in the fall of 2022. Lastly, I would like to restate that there was a decrease in adolescent anxiety and depression symptoms, but an increase in stress due to the change from online learning to in-person learning. To stem from this research project, some future research ideas are 1, trying to find ways to help cope with the elevated levels of stress, 2, surveying the same population again to see if the students had gotten used to in-person learning, and then 3, surveying other demographics such as age, specific gender, and areas in the world, etc. To also combat the safety requirements in order to complete this project, I made sure that all the respondents' questions were confidential, so no personal information was being asked from them regarding their names, phone numbers, email addresses, etc. Lastly, I would like to give a couple thank yous. I want to give a thank you to my mentor, Dr. Mandy Newman, who is currently getting her PhD at Columbia University. Unfortunately, she's not with us today because she is in New York getting her PhD, but she is watching this, so hi, Mandy. I also want to give a thank you to the Marine Chem class of 2023 and the Bellarmine Prep student body for allowing me to survey them twice. And then I also want to give a thank you to Mr. DeGroot and Mr. and Mrs. Nielsen for taking us on the Hawaii trip and also just being a part of all of this for the past 4 years, and then my friends and my family. And then these are my references, so. Do you guys have any questions? Yeah. I think the question was the data from the graph first or second survey? That was from the second survey. I didn't add my first survey up there because I kind of already gave like a general talk about what it was, and I had a lot of data from that one. But basically, the findings from that survey was just that there was all very high levels of stress, anxiety, and depression being recorded back in the summer of 2021. The question was, why do I think stress increased in the second survey? I was actually thinking about this. I think stress increased because it was recorded during the school time, and in my first survey, it was recorded during the summer. So therefore, some students may have been stressed out with schoolwork or college apps or preparing for the PSATs or SATs, which is something they probably weren't worried about back in the summer of 21. But, yeah. Yeah. Yeah, I did try and get results from like Curtis or other like local schools near Bellarmine. Unfortunately, their principal did not respond to me, so I had some trouble getting some data from there. There might be some differences based on like Bellarmine schools and then like private and public schools, just based on like stress, depression, and anxiety because of like the different homework loads, I'm guessing, or just like, I don't know, just private and public, but yeah. No comment. Okay, next question. Philip? Yeah, okay, so the question was why I chose this to be my project. So I actually started developing this project back like freshman year. It wasn't really like marine chem related. I just kind of wanted to like see the differences between like all the different subcategories of like stress, anxiety, and depression within my high school. It was also being developed like during COVID, so I was also dealing with a lot of these like mental health symptoms, and I know that a lot of my friends were. So I just kind of wanted to do something that was applicable to me and like everyone else at the time. Yeah. Yay.
All right, well, hopefully you can all hear me. Hi, my name is Anna Matson, and I guess I'll wait for this to load. Hopefully it will. There we are. What? Oh, I don't have any puns. He's saying that because Ben earlier was trying to get me to make puns about octopuses. So maybe when we're in a break, Ben will make some good octopus jokes, I would hope. Anyways, my project was on the determination of internally textured artificial DEM preference for octopus rubescents, also known as red octopuses. So to preface this a little bit, I want to show you or highlight these two maps. So this map is the distribution of O. vulgaris, also known as the common octopus. So you can see that they live on the coast of pretty much everywhere in the world, hence the name common octopus. And the octopus that I worked with, known as O. rubescens, or the red octopus, lives along the west coast of the Americas. And so the reason that I'm highlighting these maps is to say that there are many different species of octopuses around the world, and their habitat is all being affected. So there has been habitat degradation, which is affecting octopus population, declining the populations. So some factors that we're doing this is global warming. Obviously, there's ocean temperatures rising with the CO2 being absorbed into the water. This has especially affected sea surface temperatures, which is the majority of habitat where octopuses occupy in the ocean. Another factor declining populations for octopus species is the lack of fishing management. So especially for O. vulgaris, or the common octopus in the Mediterranean right here, there's high market demand for fish and octopus especially, and so there is really a lack of fishing regulation for octopuses and fish in general, so population decline has been huge in these areas. So why does this all matter? Well, octopus are keystone species. They're species that are both predator and prey, so their flux indicates problems in the environment. Octopuses have also been used to look at biomedical research because they have extremely high neural function and biomechanics. So octopuses are extremely important. To prevent octopus declines, artificial dens are a tool that have been used to combat this. So what exactly are artificial dens? Well, artificial dens mimic natural octopus dens in the wild. They are typically made out of plastic or concrete. They're usually a cylindrical or vase shape. They provide habitat. They have been found especially beneficial for brooding females. A brooding female, I'll refer to this a few times, is an octopus who has laid eggs and is then protecting them in the den. So because brooding females have been especially found to like these, this is good for their populations. So many of these tests, these are all previous tests that have been done. They have been in the wild and they have been done on O. vulgaris because it is the common octopus and the most known and studied. Because all of these past studies have looked at artificial dens in the wild, I wanted to focus my project on optimizing the use of these dens rather than whether these dens actually work, because it has been proven that they do work and octopuses do use these in the wild. So the hypothesis that I was testing was that octopus rubescents would prefer dens with a rough internal texture compared to a smooth internal texture due to their natural rocky substrate environment in the wild. So to do a little more background, the octopuses that I was working with were collected in Admiralty Bay up near the San Juan Islands, and then my actual study was conducted at the University of Washington Friday Harbor Labs on San Juan Island in Friday Harbor. These pictures over here on the right, sorry you can't see the pointer, are the pictures of my mentor Willem diving and getting the octopuses from a bottle field. A bottle field is basically a collection of glass bottles in the ocean where there's an accumulation, and the octopuses love to live in them. So there's a picture, you can clearly see the octopus in it, and then my mentor Willem. So to begin my project, I individually made each den in Bellarmine's pottery shed on the pottery wheel, and I threw each one myself. So there are two textures that I made, the rough internal texture and the smooth internal texture. The rough internal texture was made using metal clay tools to rough up the inside and mimic rocky substrate, and the smooth was just made and then dipped in clear PZN glaze to minimize bias due to color. So octopuses have been before seen to show bias towards colors, so I wanted to make sure that this was not an issue. Each den was about 14 centimeters long, the opening was 8 centimeters wide, and the biggest expanse was about 10 centimeters wide, and they were 1 to 2 pounds. Both types of dens were double fired and then soaked in distilled waters bidaily for about 10 days to prevent any chemical leaching when put into the tanks with the octopuses. So my octopus sample, I had initially 8 octopuses, but then I ended up only using 5 of them. As I said before, they were collected in a bottle field. The reason that a few of them weren't used, one ended up being a brooding female, the other one was deemed too old, and the third actually happened to be a GPO, or a giant Pacific octopus, that was very small. So these are my 5 reds that were included in my study. They were all checked out and seen to be healthy, and they were acclimated to their tanks for about 20 days before I started my actual research with them. So this picture right here is our lab setup with each tank. It was a 4-quart storage container with latching handles and bricks, so they couldn't escape because octopus are very smart and they tried many times. This picture over here is how each individual tank was set up. So the rough dens were marked with a red dot on the bottom, and the green dens, or the glazed dens, or smooth dens, were marked with a green dot on the bottom, and then rocks and sediment were used to fill in the rest of the area to mimic natural environment. They were placed parallel in the back of the tanks, so each tank was set up exactly the same. There was tank flow. Friday Harbor Labs has a special tank flow system or pipe system where water from the ocean is pumped through and filtered. So each den had fresh water running through it and was being monitored by us. So the tracking period was about a 25-day period from July 3rd to the 28th. Each day I checked them around 11 a.m. This is due to the fact that octopuses, or many octopus species, are nocturnal. They were marked in 4 categories: glazed, rough, active, or in open water. So open water was the option where they were sleeping but in neither den. So here's my results. Figure 1 shows the amount of den type inhabitants each day over the 25-day period. So this axis shows every single day that we were collecting, and this axis is the amount of octopus in each den according to this color key. One key thing to notice is all of the orange, which is the majority of what's taking this up, which is the texture. The octopuses inhabited over half of the textured dens for 84% of the days. The octopuses on average spent 12 to 24 days in the textured dens versus 1 to 6 in the glazed dens. So this graph is comparing the average amount of octopus each day in each category. So the categories, again, are water textured, glazed, or active, and water is, again, open water. So the data collection showed that the textured dens were inhabited for more than 50% of the days, whereas the glazed dens were only inhabited for about 20% of the days. This is in agreement with the average amount of octopus in each den per day. So it was a little bit over 3 for the textured dens and less than 1 for the glazed dens. This data I ran through a chi-square test, which is a test to see whether the data is statistically significant between categorical and numerical variables. So my data had a p-value of less than 0.0001, so it was in fact statistically significant, seeing that the bar to pass is 0.01. So my results did support my hypothesis. They did prefer the rough to smooth dens. They chose the rough dens 62% of the time, compared to the glazed dens, which they only chose 15% of the time. Previous research like this has not typically been done, but there have been a few studies looking at octopus habitat specifically and what kind of substrate that they like. So in Guerra and Liete, they both were found to like hard or rocky bottom substrate, which for reference, hard was described as cobble or bedrock and outcrop, and then the smooth bottom substrate was described as sand. This is another important graph to point out. So I had an outlier in my data, which was Sauron, or Orubescens 2, who spent most of its time in open water rather than either of the textured or glazed dens, which they spent their equal amount of time in. So octopuses are theorized to be migratory creatures. There have been a few studies about this, like Simmons et al. So they are supposed to be regularly adapted to changing dens. So we theorized, me and my mentor, that Orubescens 2 not being able to change from their initial glass bottle den into my den was likely due to stress or confusion. However, even though I had an outlier in my data, it was still statistically significant. So some limitations that I had a small sample size, but as I said, this didn't super matter because my data turned out well. There was limited variation on the den gradient of texture. My initial study was based on this one at the top, where I had 3 textures of den: a smooth one, a rough one, and a plain one. But due to the tank size, it varied due to also the size of the octopus. So they would rest like this in a few of the tanks, which created bias, which obviously we did not want. So I removed the plain, and I was just comparing the 2 extremes of the rough versus smooth or glazed dens. Another thing that people could go in and study besides den gradation, which I would suggest, is scaling the habitat to other species. testing it in the wild. You could do a variation of things with the dens themselves, like color. As I mentioned, octopuses do react to color, texture, material, shape of the actual den, which I was considering doing at first. And for my thank yous, I would like to thank my mentor, Willem, who sadly is not here tonight, Ariel and Lucy, who were 2 undergrads also working at the lab, doing a different project on octopus attention span, University of Washington, Friday Harbor Labs for letting me do my testing there, Mr. Nielsen for being a great mentor, Mrs. Nielsen for being the best van driver, Mr. DeGroat for also being a fantastic mentor, my mom, dad, and sister, and of course, the Marine Chem class of 2023. And there are my references. Does anybody have any questions? Yes.
So you described the four different things for in the water, in the 2 different dens, and then active.
Yes.
How is active different?
Well, active means they weren't sleeping. So I was looking into where these octopuses would take their habitat, which is basically where they want to reside when they sleep. So there was only one point that I had in my data where it was active, and that was just by chance that the octopus at that point was not asleep. So yeah. Anything? Oh, there's a question way in the back. Was it? How long was the study for? Yeah? Oh, it was, I think I said 25 days, July 23rd to the 28th. Yes. Yeah.
Can you go back to the slide where it has like the 5 parts?
Sure. This one?
Yeah. So I was just wondering, like, how do the octopus get their names? How did
Oh, okay. So all of the octopus are named after Lord of the Ring characters, which.
Did you name them, or like was that like they already were?
Well,
What was the decision?
so the decision behind this was me and the 2 undergrad girls at the lab named all of them, and my mentor Willem, of course, too. We all decided on the names together. Yes. Mr. DeGroat. These octopus, they were released back, although the red octopuses do not have a super long lifespan, so I would honestly guess that they are likely not alive. I don't think so. I think they were just put back in Admiralty Bay. Yes. Did I have a favorite octopus? I did. It was the GPO, Lady Gollum. She wasn't in the study, but she was very fun and quite mean at sometimes. Yes. Anything else? All right. Thank you.
Hello, I am Katie Sue, and I did my project on testing the rate of growth of different Phytophthora species on otter plants. So, Phytophthora is a plant pathogen that is classified as an oomycete that causes millions of dollars of damage in agriculture each year. They were first discovered in southern England and East Wales in 1993, and they spread through water to the plant's roots, and they are especially dangerous as they can seek out hosts at a small distance. Here, I have a microscopic image of the Phytophthora colony I re-isolated. Otter trees were recently discovered to be dying from an unknown cause at Titlow Beach. Otters are important to the ecosystem as they contribute greatly to providing habitats for smaller animals, supplying nitrogen to the soil through leaf decomposition, and stabilizing soil near streams. The otter trees at Titlow showed signs of Phytophthora infection, root rot, dieback, and cankers. Phytophthora was a main suspect as Titlow Beach has an aquatic environment that is suitable for Phytophthora spread. Last year, 2 Phytophthora species were found by Bellarmine Marine Chem students near the soil of these dying otters. These species were Phytophthora cambivora and Phytophthora siskioensis. Phytophthora siskioensis was first discovered in southwest Oregon on rhododendron. The host range is not well known, however, in 2009, they have been found in a grove of Italian otter trees in California. In another study, it has also been found that siskioensis is pathogenic on red, white, and Italian otter trees. I was not able to find any information regarding the discovery of Phytophthora cambivora on otter trees, but they are primarily found in noble fir and are known to be aggressive on beech trees. Phytophthora pini is already known to be aggressive on otter plants, and my samples were taken from the Washington State University nurseries as a baseline comparison for both Phytophthora cambivora and siskioensis. The purpose of the study is to test the aggressiveness of different Phytophthora species based on how fast the lesions grow on the stem of otter plants. I hypothesize that there will be a difference between the treatments, and Phytophthora siskioensis will be the most aggressive. As for my methods, 80 otter plants were first taken from the Washington State University Research and Extension Center to the Bellarmine Prep Greenhouse. They were kept in the greenhouse for a few days, approximately 2, before they were moved outside because of the temperature. After a few weeks, I moved them back inside to be inoculated. During inoculation, the plants were cut approximately 2 centimeters, about 1 foot above the roots. The Phytophthora was then placed inside the cut and wrapped with a strip of wet paper towel and laboratory film. The tweezers and scalpel used to handle the Phytophthora were sanitized with 70% ethanol between each treatment and trial. For the next month, the plants were watered every 3 days for 20 minutes at 8 in the morning. I began to measure the lesions 1 week after inoculation, continuing data collection for the next month every Monday. Interesting symptoms were also noted down. After 6 weeks of data collecting, I had to re-isolate the Phytophthora to confirm the cause of the lesions. The plants were cut approximately an inch above and below the lesions and taken back to the Washington State University. There, half-centimeter pieces were cut from the lesions and washed in bleach. The stem pieces were then rinsed with water twice before being inserted into the petri dishes containing V8 agar. The dishes were checked the next week for Phytophthora. The data collected on the length of the lesions were analyzed using a ANOVA and post-hoc tests to see if there were any differences between the treatments. On my slide here, I have the average length of each treatment with error bars on the final day of data collection, which is October 31st. Each treatment contained 10 plants, and there were 2 trials. So this graph shows the average of 20 plants for each of the treatments. Looking at the graph, Phytophthora siskioensis had the highest average length at the end of data collection, with Phytophthora pini in second and Phytophthora cambivora in third. Through analyzing the data with the ANOVA test and post-hoc test, there were no noticeable differences between siskioensis and pini. They were equally aggressive. This agrees with the error bars on the graph, seeing as there is a noticeable overlap between siskioensis and pini. Through the ANOVA test, the data shows that both siskioensis and pini are more aggressive than cambivora. On my table here, you can see that the p-values of most combinations are close to 0.01, which means that there's a noticeable statistical difference. After analyzing the length of the lesions through the ANOVA and post-hoc test, Phytophthora siskioensis and pini are both more aggressive than Phytophthora cambivora. But this supports my hypothesis since the 2 species of Phytophthora collected at Titlow Beach are different, and Phytophthora siskioensis is more aggressive than cambivora. However, I did not expect siskioensis to be equally aggressive as pini, as Phytophthora peony was included in the research as a comparison. This might mean that Siskioensis is a serious threat to alder plants. When analyzing the data, I also had to omit the first week of data collected due to inconsistencies with the rest of the data. This might have been caused by different methods of measuring the lesions. I had to decide where to start measuring and when to stop. Some of the lesions also didn't grow continuous like the one shown here, and I had to choose whether to subtract off the unbruised areas. There were many limitations when measuring the data. Many of the plants during inoculation had part of the stem completely cut off, like the one shown in the picture here. Most of these ones have no growth at all. Many other plants did not have their lesions grow past the cut and instead started growing horizontally, which I also included on the slide here. The horizontal growth was not included in my data analysis. Many of the plants whose lesions grew horizontally at an incredible speed died quickly when the Phytophthora basically cut off the top part of the plant after the lesion completely wrapped around the stem. This most likely affected the data as I kept the length of the lesion the same for the remainder of the data collecting period. Phytophthora species mainly thrive around temperatures of 20 to 25 degrees Celsius, which is around 77 degrees Fahrenheit. However, the temperature during the first week of inoculation averaged around 90 degrees Fahrenheit or above in the greenhouse. There was no temperature control in the Bellarmine greenhouse, and as a result, the Phytophthora may have been affected. When I split the average length of the final day into two trials, there are also some interesting trends that are seen in the graphs. In trial 1, though the length of peony stays relatively the same, Siskioensis is much shorter, making peony have the highest average lesion length. However, the error bars still overlap, meaning the two species are still equally aggressive. On the other hand, in trial 2, Siskioensis has a much higher average, which looks similar to the graph that averaged the two trials, except the error bars don't overlap, meaning in trial 2, Siskioensis is the most aggressive species, with peony in second place. Here is both trials on the same graph for comparison. While I was researching, I was unable to find past studies on aggressiveness on alder or any of the Phytophthora species I was studying. I was able to find records on discovery of Phytophthora in the Pacific Northwest, the most common being Siskioensis, but many other species were also found on the same plant. This leads us to possible further research. A further study might be testing if different combinations of Phytophthora might affect aggressiveness or growth of a Phytophthora. Temperature and seasons may also impact the spread of Phytophthora, although in this area of study, there is already more information on. To also get more reliable results from our study, more trials are needed, especially to confirm the aggressiveness of Phytophthora siskioensis, as the two trials yielded different results. For my acknowledgements, I would first like to thank my mentor, Dr. Elliott, for guiding me through every step of my research project. I would also like to thank Ms. Savage for allowing me to use the Bellarmine greenhouse, supplying me with pots and soil, and also helping me set up the irrigation system. Shout out to Amaris Osted for helping me collect data and inoculate the plants. And then obviously to Dr. DeGroat, Mr. and Mrs. Nielsen for being great mentors and taking us to Hawaii. And obviously the Marion Chem class of 2023, especially a certain man. And then finally, my parents who have supported me on this journey. Thank you so much. Are there any questions? You in the back? The question was, could it be possible that the water quality affected the growth? And I think it might have, but considering that my irrigation system connected both tables to the same hose, it was not very likely it would have made very different between like all my treatments and trials. Thank you.
One, two, three. 1, 2, 3. Hey, it's a break time. We'll take just a couple more minutes. Go ahead and stretch. And then Dominic will come up and tell us about his research. Boy, Maris getting shut. Why'd you throw that out? Wow, you still eating that sandwich?
Tyler? Yeah, no, he's a slow eater.
Yeah. Makes you want to buy it. Okay, y'all good?
Yeah.
That's a nice suit.
Thank you.
Okay, well, let's go ahead and get her going. Why don't you ask people to go ahead and take a seat? Where you want me?
Sure, you got it.
Hey, let's go ahead and take a seat one more time.
Just waiting for this to load. Hi. Hello, my name is Dominic Lessie. My project was less of a marine chemistry project, more of an aerial physics project. It was a comparative study on the efficiencies of horizontal axis wind turbines of different quantities of airfoils. So why did I even bother studying this? When I was considering what I wanted to do for my marine chemistry project, I thought about the problems facing my generation. I knew that climate change was going to be a big problem, and I also knew that renewable energies were a potential solution. So I wanted to do a project with one of them, but which one? As cool as it would be, I cannot build a nuclear reactor, so that was out, and I wasn't going to drill a 60-foot hole into the ground, so geothermal wasn't it either. I briefly considered solar energy and just buying a panel online, but I wanted to be more involved in my project than that, and besides, I didn't even know what I'd want to do with it anyways. So I landed on wind energy, and I decided I would build a turbine myself. Also, as I moved further into my project, I discovered just how much physics was at play, which was great for me, because that's one of my favorite subjects. It is a fact that the cause of climate change is the human emission of greenhouse gases. It is also known that climate change causes an increased frequency of extreme weather events such as floods, droughts, intense rain, and severe heat waves. 25% of all U.S. emissions come from the generation of electrical energy by the use of fossil fuels such as natural gas and coal. Wind energy is a potential solution to this problem and should be researched because it can provide energy while not producing emissions and because wind is an abundant and inexhaustible resource. It is also impossible to overuse the wind. Additionally, it already provides over 9% of the electricity nationwide and more than 30% of the energy in 5 U.S. states, those states being Iowa, Kansas, Oklahoma, and both North and South Dakota. There is without any doubt a clear and obvious demand for wind power. The blades of a turbine are particularly important to the efficiency because they are what catches the wind and creates the spinning motion. I noticed in my research that almost all of the turbines I saw had an odd number of blades. I learned this was to prevent one blade from sticking straight up while another sticks straight down, which can create vibrations that are harmful to stability. Even more specifically, I noticed that 3 blades was the most common configuration, so I wondered if it would be less efficient to have more. In terms of general previous wind turbine research, the first thing to mention is the Betz limit, which is the theoretical maximum energy conversion rate for a horizontal axis wind turbine, which is exactly 16/27, or around 60%. This is important because it guarantees that no matter how well designed any turbine is, it will lose at least 40% of the kinetic energy of the wind. There exists much scientific literature on the topic of wind turbine blade qualities. The same cannot be said for wind turbine blade quantities. However, a study I found specifically on blade count, TAN 2016, discovered a trend that turbines with more blades performed better in slower wind speeds than their counterparts did with fewer blades. So I hypothesized that in slower wind speeds, a 5-bladed turbine would be more efficient, while in some higher wind speed, a 3-bladed turbine would surpass it in terms of efficiency. So in my own driveway, I got straight to work making a total of 8 blades for my turbines. The blades were cut from 1/8-inch thick PVC, that is polyvinyl chloride, piping. PVC was chosen for being lightweight, cheap, and requiring relative ease in getting it to resemble the blade of a wind turbine. Power tools were used with caution to divide the piping into 90-degree fourths, which were then shaped even further with the use of a table saw. The end result were 8 blades, each 35 inches long, with an average mass of 256.5 grams. The next part of the turbine to be constructed was the rotor. The rotor is the central hub upon which the blades attach. It is essential to the balance of the turbine that the rotors were crafted with precision and accuracy. For this reason, the rotors were designed in Fusion 360, then cut out on a CNC, that is a computer numerical control router. The end result were 2 1/8-inch thick, 270-gram aluminum plates with a radius of 4 inches, leaving plenty of space for the blades to attach. A system of pillow block bearings and an aluminum axle was used to support both turbines. The finished assemblies are pictured here outside of the Bellarmine greenhouse. I was originally going to test these turbines inside, however this provided its own challenges, so testing took place outside of Bellarmine's greenhouse. In each of the 31 trials, an iPad camera was used to record the turbine, and an anemometer, pictured on the right here, was used to determine the wind velocity in each trial. After each trial, the video would be analyzed to determine the angular velocity and the given wind speed. Other variables such as temperature, dew point, and air pressure were determined online and then used in later calculations. Rotational inertia will be important 2 slides from now, but it can just be thought of as how easy or difficult it is to spin an object around a fixed axis. For the purposes of this presentation, you don't need to know how exactly I got these rotational inertias, just know that they are dependent on the mass of the spinning object. So the 5-bladed turbine had a greater rotational inertia compared to the 3-bladed, slightly less rotational inertia. And these are my results. Here we can see the angular velocities of each turbine in the range of wind speeds tested. The relationship can be approximated to be linear and positive. Here we can observe the rotational kinetic energies of both turbines as a function of the wind speed. This is an exponentially positive relationship. It is also why it was necessary to determine the rotational inertias up in that equation in the top right corner. The next graph is of efficiencies and different wind speeds, but I figured it would be helpful to define what efficiency is before showing these graphs. Efficiency is the kinetic energy the turbine spins with divided by the kinetic energy of the wind blowing at a given constant velocity over a defined two-dimensional area at a constant air density for 1 second expressed as a percentage. Also shown here is how that kinetic energy is defined and calculated mathematically. And here are the graphs. This is what they look like. From the data collected, the most clear and predictable trend was that both turbines spun faster when in faster winds. This makes perfect sense. Moving on to something with more implication, it is clear from the data collected that the relationship between wind speed and rotational kinetic energy is exponential. It is of the utmost importance that wind farms are built in the windiest places, not the second most windy places, because it being just a small amount less windy can mean significantly less energy for the turbine. Thirdly, there are some problems with the efficiency graphs that should be addressed. For one, there's a point that breaks the Betz limit. On the 5-bladed efficiency graph, we can see that there is a point, this point, which is above 60%, which should be impossible according to the Betz limit. The likely explanation for this is human error. It is possible that a faster wind blew for some time, causing the turbine to spin faster. The wind then died down, but the wind turbine is still spinning quickly, and it was at that moment that data collection took place. This would yield a data point which would appear as though it broke the Betz limit. Secondly, these graphs are just kind of messy, but they can be fixed with a small amount of algebra. It has been established that angular velocity is dependent on the wind's velocity. Therefore, angular velocity is a function of the wind's velocity, and because it is linear, bust out your TI-84s, because we can use linear regression. This process reduces the effects of small imperfections in data collection by reducing the number of variables down to 1. This process reveals much clearer trends. My original hypothesis that the 5-bladed turbine would be more efficient in lower speeds but would be surpassed by the 3-bladed turbine in higher speeds was only partially supported. As we can see from this data, the 5-bladed turbine outperformed the 3-bladed turbine in every trial conducted. The additional 2 PVC blades on the 5-bladed turbine caused it to outperform its 3-bladed counterpart. But the 3-blade simply never surpassed it. The additional rotational inertia and drag force was insignificant compared to the gain and lift force from the greater area for the wind to strike. The findings of this study align entirely with the findings of TAN 2016. More blades offer superior efficiency in lower wind speeds than their counterparts with fewer blades do. Every study has its limitations, and mine is no exception. The most influential limitation was that data collection took place outside as opposed to in a laboratory's wind tunnel. This limited the range of speeds it was possible to test in because the wind velocity was entirely dependent on the weather of that day. Being outside meant that on different days of testing, the air density was slightly different. This difference can be treated as negligible, but it is worth noting for the prospect of future studies. It is recommended that, if possible, future research can be conducted in better controlled settings. In terms of future research, at what point does adding PVC blades stop adding efficiency and start creating inefficiency? Also, how could the results of this study have been different if the blades used were substantially larger, more similar to the blades on turbines in wind farms? Another factor to consider is economics. Turbines in actual use have larger blades made of materials more expensive than PVC. This prompts the question, if the optimal number of blades is greater than 3, which my data supports, is the difference in efficiency they provide worth the additional cost in manufacturing, or would it be better to save on the cost of 2 extra blades and construct a second 3-bladed turbine? The answer to this question may lie in knowing the specifics of the wind turbine in question. In essence, more research is needed. Lastly, something more to consider are vertical axis wind turbines, which are exactly what the name implies. I chose not to study them because they see less use and are simply more complicated, but they would make for an interesting potential future marine chemistry project. First off, a big thank you to Mr. DeGroot and Mr. Nielsen for guiding me during my project and for the amazing trip to Hawaii. Also, a thank you to Ms. Nielsen for chaperoning as well. A thank you to Ms. Savage for lending me space in the Bellarmine greenhouse for my project. A thank you to Cole Wicklander for helping me with the use of the CNC machine for creating the rotors. And last but not least, thank you to the marine chemistry class of 2023. These are my sources. Were there any questions? I see one in the back. Save? you get a really big fan. The trouble was that the turbine was bigger than any fan I could find. So when I was trying to calculate the swept area, hold on, in this equation here, the A is for the swept area of the turbine, and the wind created by the fan wasn't big enough to hit the entire turbine, so I had to go outside and use the actual wind. Mr. Nielsen? The question was, can you actually generate power for the greenhouse with this? The answer is yes, just the problem is scale. There's not enough, once you get up to speeds, the efficiency is so low, not low, but there's not enough energy to actually do anything with it. If we look at the kinetic energy graph here, it's only 10 joules, which is 10 watts, because a watt is a joule per second, so it's really not that much energy. Any other questions? No? Thank you.
All right. Technical difficulties. All right. Just a moment. Okay. Hi, my name is Corinne Harrison, and my project was evaluating the prevalence of methicillin-resistant Staphylococcus aureus, or MRSA, on an artificial turf field. I was first inspired to research artificial turf when I heard the incredible story of Alex Smith. As a player in the NFL, he suffered a multiple compound fracture to his leg during a game. After surgery, he was told he would make a full recovery. But a few days later, he was in septic shock and at risk of losing his leg. He had acquired a bacterial infection from the artificial turf on the field, and it had been closed inside his leg during surgery. After multiple surgeries to remove the dead tissue and recover what was left, he was told he would never play football again. While Alex Smith's infection wasn't MRSA, artificial turf fields have been known to host a greater abundance and diversity of bacterial growth than natural turf fields, because rubber or sand turf infill, as well as turf fibers, create excellent hiding spots for bacterial colonies. Contact sports such as football, soccer, and lacrosse often involve intense player contact with turf, so when open wounds are exposed to turf, they can quickly become infected. This makes MRSA the leading cause of bacterial infections in high school and college football, as well as other contact sports. Now, why MRSA specifically? It's not a total mystery. MRSA has been shown to reproduce at a rapid rate when exposed to a chemical known as mucin. That's a natural substance found in mucus and saliva. And we all know how soccer, lacrosse, and football players are known to spit on their fields. MRSA exposure can cause, gross out warning, blistering skin infections. In more severe cases, it can lead to serious infections of the blood, heart, bone, joints, and central nervous system, or may cause a condition known as necrotizing fasciitis, more commonly known as flesh-eating bacteria disease. MRSA is also resistant to most antibiotics used to treat infections of its nature. So my research question was, how prevalent is MRSA and where is it most prevalent in the artificial turf on Memorial Field? Specifically, I predicted the following. MRSA abundance will decrease with time elapsed since varsity football games. MRSA abundance will be greatest at the center of the field and will decrease with distance from the center. MRSA abundance will increase in samples taken at cold temperatures, and MRSA abundance will increase in samples taken at high humidity. To examine the abundance of MRSA and other microbiota on artificial turf fields, I chose to perform a series of hardy-chrome MRSA tests. Traditional bacterial cultures grow high rates of pathogenic and non-pathogenic Staphylococcus species that closely resemble MRSA and would have required significant bacterial culture workup. Hardy-chrome MRSA agar plates inhibit most bacterial growth and therefore reduced the demand on my time. They're very convenient. I measured ground temperature using a VG scientific soil thermometer and measured air temperature and humidity data using a ThermoPro TP49 digital hygrometer and thermometer. Hardy-chrome MRSA agar plates and standard medical grade rayon swabs were used for inoculation. And this is what a MRSA colony looks like on hardy-chrome plates. Now, in order to investigate the effect of 3 varsity football games, the highest attended games on Memorial Field on MRSA growth, I tested the field shortly after those games ended, the day after the game, and 3 days after the game. Here you can see all 9 sites. We're viewing the field from the home side. It's 3 at either 30-yard line and 3 on the 50. I used 2 swabs for testing at each site, inserting them approximately 1 inch into the turf 10 to 15 times and inoculating them in parallel on hardy-chrome MRSA agar. I repeated the process for the second swab on the other half of the plates, replaced and labeled the lids, and transported them back to the laboratory for incubation. Now, this is a lot of data, but I performed a total of 81 turf cultures during the study period. 13 specimens were positive for MRSA. That's a 16% positivity rate. 38 were positive for methicillin-resistant Staphylococcus intermedius, a type of staph bacteria that grows on the skin of dogs and cats. And 27 were positive for unidentified bacterial growth. But let's put this in a format that's a little easier to see. There we go. That's all my data graphed by collection date. First, let's examine the effect of time on colony growth. The data I collected suggests the time elapsed between varsity football games and the number of MRSA colonies collected have very little correlation. The average number of MRSA colonies collected remained essentially the same whether the varsity game was 0, 1, or 3 days before collection. Similarly, the average number of colonies of Staph intermedius did not appear to be correlated with time elapsed from varsity football games. Unidentified bacteria typically experienced a large population spike after 3 days. All right, let's move on to the effect of location on bacterial growth. Here is my data for all 3 types of bacteria. As you can see, MRSA was the rarest of the 3 types, but this heat map more clearly shows where MRSA grows on the field. Again, we're viewing Memorial Field from the home side. The data suggests MRSA is most abundant at the south side of the field, right over here. Sites G, H, and I repeatedly showed high levels of MRSA and other bacterium, suggesting there may be some correlation between the sampled area of the field and the presence of resources for MRSA colony growth. It also appears to be slightly more prevalent on the visitors' side, so interpret that as you will. The 2 other types of bacteria aren't shown here, but Staph intermedius grew most frequently at sites B and G, and unidentified bacteria grew the most at site B. Let's take a look at one of the plates from site B. All those little spots are unidentified bacteria. That's kind of gross, right? All right. I also evaluated the effects of humidity on colony growth. My findings suggest that MRSA grows most abundantly in environments ranging from an approximate 45% to 55% relative humidity. Consistent MRSA levels were recorded below 45% humidity, and one sample round taken at 60% humidity with 0 MRSA colonies shows a probable limit to MRSA growth. Now, humidity data was only taken at the time of sample collection, so it's possible that other humidity data from times before testing may have impacted my data. But my findings at sample collection, there appears to be a correlation between humidity and colony growth. Let's move on to air temperature data. The data suggests that MRSA colony growth may have some correlation to atmospheric temperature at the time of sample collection. A sharp decrease in the number of colonies, right there, was observed between 16 and 19 degrees Celsius. All right, this is the first study using hardychrome MRSA agar plates to characterize the microbiota of artificial turf. This was also an observational study, so a major limitation is that it should not be used to draw conclusions about the causes or origins of observed microbiota. But the observed data suggested three things. First, that MRSA abundance was highest at the southern side of the field. Many factors differentiate the northern and southern sides of the field. For instance, the southern side is farther from the high school, closer to a forest, and closer to the highway that runs behind it. Therefore, it may be exposed to slightly different athletes, workers, and atmospheric pollutants, or may even be experiencing reduced infill drainage. The second major finding was that MRSA abundance is highest within a range of 45% to 55% relative humidity. A previous study by Chandra Sahu found that community spread of MRSA is greatest between 55% and 78% relative humidity. I tested the field in September and October when humidity levels were relatively low, so not enough data was collected to observe the effects of humidity between 55 and 78%. Future research should be conducted on Memorial Field during high humidity seasons to determine the most accurate humidity range. Lost my train of thought there. Lastly, one major finding of this study was MRSA abundance was consistently highest in samples collected at temperatures below 19 degrees Celsius. The same study by Chandra Sahu suggests that higher temperatures are usually associated with high MRSA growth, so this study contradicts those findings and suggests that low temperatures may be ideal for MRSA growth. Future research into MRSA and artificial turf might focus on the effects of humidity on colony growth. As I said, I did not have a lot of data on weather outside sampling times, so a study focusing in more depth on atmospheric data's effect on MRSA may be of scientific interest. I also suggest future research be done on MRSA in indoor environments like basketball or volleyball courts. While artificial turf can be a great home for bacteria, indoor courts may be of interest due to their tightly controlled atmospheric environments. Finally, I believe an experimental study into possible cleaning agents may be of interest to determine the best way to eradicate MRSA from our field and protect athletes. All right. I want to thank Mr. DeGroat for all of his help and feedback. I also want to thank my parents for supporting me through all 4 years of Marine Chem, as well for driving me to and from campus and standing out in the cold while I swabbed the field. Another big thank you to the campus security staff who let me into the field on weekends and after football games. Sorry for knocking on your window with a thermometer when I got locked out. Finally, thank you to Mr. Nielsen and the real Neilmobile for being the best van crew in Hawaii. You guys rock. And thank you to the whole Marine Chem class of 2023. You guys are awesome. Okay. Thanks. Any questions? Yeah, Levi?
Yeah, so I was wondering about, it showed on the third day after the football game, there was a big spike in the unidentified bacteria. Why do you think that might be?
So the question was, why do I think there was such a big spike in unidentified bacteria 3 days after the football games? I'm really not totally sure, but every time I tested on the third day, there was always a big spike in bacteria. In this study, I didn't account for girls' soccer practices, which were also going on at the time. I almost got stuck in the middle of a girls' soccer practice after school one day. So that may have had some effect on it. I know their games are on a slightly different schedule. But that unidentified bacteria, I can't determine what that is or where it came from without doing, Madeline's type of DNA workup. So, all right. Any other questions? Yeah, Mr. Nielsen? Mm-hmm. Mm-hmm. Yeah, so the question was, what's the assessment when a player gets a cut on their leg or gets, a cut of any type on the field? I know that in the academic training room, they're trained to deal with those sorts of things. Usually on fields, there's some sort of medic to deal with that. I have heard of other instances of getting MRSA off the field, so in gymnasiums indoors. It can come from all over the place. So it comes from everywhere. MRSA is everywhere, and it's very scary. MRSA isn't completely resistant to all antibiotics, though. It does respond to some antibiotics, so there are passive treatment. Okay. Any other questions? All right, thank you.
All right, hi everybody. My name is Evan Jenikiewski, and my project was on image classification for crab zoe and megalope larval stages using machine learning. To begin, what is machine learning? Machine learning is a subdivision of AI, which is artificial intelligence. Artificial intelligence attempts to replicate or mimic the human brain and how it functions. And machine learning, more specifically, is the ability for a computer to learn and adapt without instruction. This is done through pattern recognition. This pattern recognition is seen throughout our daily lives, whether it be song recommendations based off your previous listening, identifying individuals in your camera roll, or even something as simple as autocomplete on Google. Why did I choose crab larvae as the subject of this study? Crabs are a glimpse into the health of the marine ecosystem. The Washington Department of Fish and Wildlife monitors adult crab populations through the use of catch record cards by fishermen. And these adult populations allow estimators for other organisms and their populations to be made. However, there's little data on crab larvae. Crab larvae not only offer a glimpse for estimations of other organisms' populations, but also for future adult crab populations. There are two types of crab larvae in which we will look at. The first being zoe. In the image, it is labeled as A. And the second is megalope, which is labeled as B. The crab labeled as C in the image is a juvenile crab, which will not be looked at in this study. Machine learning has gained much traction over the past years and is very useful for data gathering and data analysis. It's very beneficial for researchers as it helps automate tasks and can help make things more efficient. The objective of my study was to create a neural network that can identify the zoe and megalope larval stages of crab. There are two major sections to my methods: the collection and processing of the crab larvae, and the programming and the training of the neural network. I collected the crab larvae at Point Defiance Marina. There's an abundance of crab here, and there was also access to a dock. After gaining approval from the park, I was able to set up the light trap, which is used to collect the crab larvae. The light trap came from a previous Bellarmine study, so I did not have a need to construct it. It consists of a watertight box that sits on top of the floats, and this box holds the battery, which powers the green LED light. The LED strip hangs down below the float and connects to a bucket that also hangs below the float. This bucket has some modifications to it. It has holes drilled on each side of it with funnels that allow organisms to swim into it but not out. On the bottom of the bucket, there's a mesh filter that allows the drainage of water but catches the organisms before they fall out. After setting up the trap by threading the LED strip into the bucket, I let it sit overnight. In the mornings, I would drain it, and all that would be left would be the organisms caught by the filter. I would separate the crab larvae from the other organisms and prepare them to be transported to Bellarmine. Back at Bellarmine, I would photograph the crab larvae. The images on the right are a photograph of the microscope, of the dissection microscope, and the Motocam camera that was used, and a picture of all the crab that was collected by the mesh filter of the trap. I placed the larvae on the slide one at a time and took multiple images of each one. I took a total of 320 images, 160 of each type of larva. The majority of these larvae came from the Dungeness and red rock crab species. However, there may have been some others. These two images are an example of the two that we're taking. The left is of a zoe and the right a megalope. A zoe is characterized by the horn on top of its body and small feet below. It also has a long forked tail. The megalope resembles more of a juvenile and adult crab. However, it does have a short tail. Just in this image, it is curled beneath. Now that I have the images, the next part was to program and train the neural network. I selected Python as the language as it is the most common for machine learning. And alongside it, I used TensorFlow, a library created by Google for both beginners and experts in machine learning. The method of pattern recognition that I used was image classification. Image classification allows the computer to be given an image and it outputs what it sees in that image. So I would be able to pass in an image of a zoe and the computer would then tell me that it sees a zoe in that image. Data augmentation was also needed. Due to the small number of images that I collected, 320 is not as sufficient as it seems for a computer to learn from. Data augmentation through either cropping, rotating, zooming into, or changing the contrast of an image allows for that data set to be multiplied and increased a substantial amount. The method for training the data I used was a train test split method. The train test split method takes all the data and splits it into two major categories. You can see this in the image on the top. There's all the data, and then there's the training data and the testing data. The training data consisted of 80% of the images, both the original and the augmented ones. The testing data, the remaining 20%, is used to validate that the training was successful. I trained the model over a total of 100 epochs. Epochs are iterations through the data set. And these iterations means that each image, including the augmented ones, will be seen a total of 100 times. This training leaves us with two types of results, loss and accuracy. Loss is essentially the punishment for getting a guess wrong. The more guesses that are incorrect or wrong in each epoch or iteration results in a higher or lower loss value. The loss is ideally close to zero, and when graphed, we would want to see a negative correlation. Accuracy is a value from 0 to 1 and is essentially the percentage of correct guesses. When graphed, accuracy should have a positive correlation, and an ideal value would be 0.9 to 1. This first graph is of the accuracy for both the training and validation set. The training set, the 80% of images, is in blue. It begins at around 0.45 and increases to a value of 0.9. The validation set begins at a value of 0.6 and increases to a value of 0.9 as well. However, it oscillates more heavily than the training set. I will touch upon this a bit later. The second graph is for the loss. The training set begins at a value of 0.7 and decreases to a value of 0.2. The validation set begins at a value of 0.7 as well. However, it begins to heavily oscillate around 70 epochs between the value of 0.6 and 0.2. This is not ideal, and I will also explain this later. These are two images that I downloaded off of the internet. I inputted them into the neural network to test the accuracy of it and to show me some results. The left one is of a megalope. However, the computer was 87% confident that it was a zoe. I believe this error comes from the presence of the tail. As the computer learned through pattern recognition, I believe it began to associate the presence of a tail with a zoe, as that obviously has one. However, the images of my megalopes may have curled their tail underneath, and it is not as common to see. The image on the right resembles more of the training data that I used and is a zoe. The computer was 100% confident and was correct with its guess. Based off of the graphs and the data, I'm able to say that the model can distinguish between a zoe and a megalope. However, with the heavy oscillation on the validation graphs, I'm not entirely confident that I know that there are some errors with the pattern recognition that happened. Although the accuracy both trended upward and became an ideal value of 0.9, which means around a 90% accuracy rate, and the loss decreased to a value that was ideal, the heavy oscillation hints at common mistakes. The oscillation also suggests overfitting. Overfitting is where the model begins to memorize images. This memorization is bad. When it begins to memorize images, it begins to not guess and just know. The training model will begin to have a high accuracy rate and a low loss rate, whereas the validation will begin to regress and try to get back to an earlier state to learn more. These limitations altogether come from some of the possibilities from when I was collecting the crab. I attempted to preserve the crabs when I was collecting them, but using iodine or alcohol proved unsuccessful. This resulted in me having to take live images of the crab. The crab larvae did not want to cooperate. This reduced the types of photographs I could have. The crab megalopes did not show their tails in most of the images as I showed in the example, so when passing in the image that had 87% confidence, resulted in a lower quantity of images. The types of photographs, as I mentioned before, were also limited. I was not able to position the bodies of the crab larvae how I wanted them that would be ideal for the learning of the computer. These limitations I mentioned hint at for future research. The first one being increasing the data set. Increasing the data set would lead to a higher accuracy as there is a larger variance of images and a larger selection that it can learn from. The second area for future research is object detection. Object detection is another method of pattern recognition. I opted for image classification in my research project, however, future ones could choose object detection. Looking at the image on the left, it draws boxes around multiple cars, a truck, a person, a bicycle, and even a traffic light, all in one frame. This is what object detection allows. Object detection gives live feedback and can allow multiple objects to be identified within one frame. Applying this to the image on the right, it would be able to draw boxes around each of those crab larvae and identify them as megalops. However, with image classification, I would need to crop that image to show only one crab larvae at a time. This reduces the efficiency, and for researchers in the field, object detection would prove to be more streamlined and efficient. Another method, or another area for future research would be underwater detection. Underwater detection could be done by either using a camera set up in the trap or an underwater drone to photograph the crabs in their swimming state. Having both images of crab in their swimming state and stationary state would be very necessary in order to help researchers have more accurate counts. Researchers place organisms within a tub with a thin layer of water. If the crab larvae are swimming around or stationary, having the ability to identify them in both states would allow them to be more efficient. These areas for future research would help eliminate the need for human counting and would help make research and tallies for crab larvae on their population counts to be more efficient. Here are my references. And thank you to my family, Mr. DeGroat, Mr. and Mrs. Nielsen, and the Marine Chem class of 2023. Are there any questions? The question was, do I think this would work with other organisms too? Yes, this could be applied to other organisms. However, the training data would just have to be images of whichever organism that's wanted to be used. However, the code that I ended up programming could be applied. It just needs a new data set to be used. Solve it.
What is more than Python?
Could you repeat that?
What packages in Python?
the question was what packages in Python did I use? I used matplotlib, and otherwise it's mainly just TensorFlow. Mr. DeGroat? Yeah, so the question was, were these labeled when I took them off the internet? And yes, I downloaded them from sites where they were where the left one was labeled as a megalope and the right one was labeled as a zoe. Oh, no, I'm not sure what species necessarily they are. The question was, what inspired me to do this project? My brother did a similar project along the same lines, and I've always been interested in computer science, so I thought this would be an interesting area to pursue, and Mr. Nielsen and Mr. DeGroat gave me the idea for using crab larvae, as Dungeness crabs are important to the Puget Sound. Yes? So the question was, when I pass in an image to the computer, does it rotate it, shrink it down, or zoom in? No, not when it's learning. This is what the data augmentation is for. So it doesn't rotate it at every possible degree that it could, or crop it at every possible way that it could. It just, the data augmentation randomly chooses which ones to crop and crops them on a random scale and does that with each type of augmentation. Yes? The question was if I would share this with like the Department of Fish and Wildlife. I could share this, however, it's not a mobile application, so it wouldn't be as beneficial to them, but seeing the data, they could pursue it in an area. However, I think a private organization might end up pursuing it in more depth. Yes? The question was if the angle that the photo is taken at affects the accuracy of identifying them, such as the zoe laying on its side or megalopes standing, or as a bird's eye image. This is how they just laid down. Since they were live, I wasn't able to manipulate them as easily. The zoe naturally fell on their side and curled up as they weren't suspended in water, and the megalopes were just standing. However, if they were preserved, I could possibly manipulate the bodies to be in many different rotations, which would provide a greater variety of accuracy. All right, that's all. Thank you.
Okay, so next up, I guess we're done for tonight. We're going to be back here on Tuesday. I really appreciate you guys coming out and supporting our seniors. Really cool stuff. I'd like to give one more big hand of applause. There's a lot of, like I said, a lot of cool stuff here. You'll see it also on Tuesday. For our freshmen and for our sophomores, take note, we're really interested in continuing a lot of these different lines. And so if something kind of jumped out at you, come talk to us right now. You don't need to wait until your junior year. So, again, thanks for coming this evening. Have a safe drive. And if you want, I think